Now, when you study ecosystems, you're basically looking at the living organisms and their environment, right? Now, the findings of any ecological study must be presented in a report, right? So it must have the following heading. You must have an introduction, you must have the method. So the introduction would be like what it is you're studying, right? What it is you aim to study, right? Why you're doing this study, I should say, right? The method would be like the procedure in which you decide to carry out your sampling technique. It would also be like the apparatus that you're going to use for the study. Your results and observation must be laid out. So all the data should be presented in public tables. If you have graphs to create, to be able to make a comparison, histograms, profiles, etc. And you must be able to study the relationship and explain what you're seeing taking place in that particular environment, right? Now, the discussion must be present and observation. So you must explain, analyze the patterns, discuss what's there and whatnot, right? Now, the purpose for studying an ecosystem is to study the biotic, which is the living environment, and the abiotic, the elements associated in the ecosystem. This would be able to help you determine the relationships between the plant and the animal population, right? So it will be able to better tell you, well, okay, these are the plants you have, so these are the animals that you're gonna have. But these are the animals here. This is why these plants are in high population or low population, right? Any questions there so far?
No, ma'am.
All right. Now, when you're choosing an ecosystem to study, if you choose too much of a small ecosystem, right, you wouldn't have plenty species to actually be able to examine, right? So you choose an okay size one with enough organisms that you could examine, like a pond or a small woodland, right? Or a piece of wasteland or an area, a grassland, right? So that you have a good bit of organisms that you could compare. Not too little bit, but at the same time, not too large where you can get overwhelmed with the study, right? And then you create a scale outline map of the ecosystem, you draw it, and as the study progresses, the position with the most abundant species should be marked out on the map. Any questions there so far?
No, miss.
All right. Now, there is something called sampling methods to be able to study the organisms that are present. So different species of organisms will present themselves in the ecosystem, right? And they will have different numbers. So it will have different variety. The species will be in different amounts all over the ecosystem. So that must be determined to know, well, how much of that species is living there in that area. You can't count all. It's almost impossible, right? Because you can feel your count all and then boom, one organism show back up again, right? So we could use some sampling techniques to be able to kind of give us an average of what's going on in that area. So the first sampling technique that we could use, it's not very difficult to do, is observation. So your ecosystem, you can actually visually observe it. Look at the common species of plants and animals and list out anything. If you see certain plants or certain animals creating adaptations to survive in that environment, you make a note of it, all right? Any questions there so far?
Nope.
All right. Now, we have quadrats. Now, it's a square frame. Let me see if I can get an image of this for you guys. Hold on. Yeah, it's hot. This is one that we typically do in schools, right? I remember working with my students with this particular one already, right? So it's a square grid, right? So this is what the grid would look like. And it's placed several times at random, right? I'll play the video just now for it, right? It's placed several times at random in an ecosystem. And the number of species of plants or animals, right, that are inside of those blocks are counted, right? Now, if it's impossible to distinguish the individual plants, right, the percentage of the quadrat area they covered is estimated. So it's used to study like a fairly uniform ecosystem. So you used to study like a grassland, right? Something that don't have too much variety, right? So I'm going to play a short video here based on quadrats and the use of quadrats, and then I'll go on to the next one for you guys. Give me a minute.
Hello, this is Dr. Acton from Malmesbury Science, and this is quadrating. So today we are going to do an investigation to estimate the population size of daisies in our field. To do this, you will need a quadrat and you will need to know the area of your field. I've measured the length and width of the field using a trundle wheel, approximating it to a rectangle. Multiplying those gives me a total area of 620 metres squared. Ideally, what you want to do is to split this area into a 1 metre square grid, for which every square has a coordinate, top right being 3120. Then, use a random number generator to choose random coordinates for your quadrat. This removes any bias. To be accurate, you should aim to sample around 5 to 10% of the total area, so I should sample 30 positions, but I'm just going to choose 10 today. You then place your quadrat at those positions and count the number of chosen organisms, which in our case is our unique species of flower, Dasius papiralis. So once your quadrat is positioned, you need to count the organisms, in this case daisies, in the 1 metre square. 1, 2, 3, 4, 5. This one here is on the edge of the quadrat, but because over half of the flower is within the quadrat or underneath the quadrat itself, we will count that flower. So that means there are 6 flowers in this quadrat. We would not count this daisy here because it is not over half in the quadrat, so it does not count. Even if it is partially covered by the quadrat, it does not count unless at least half of that organism is within our 1 metre squared area. Here are my results. What I then need to do is find a mean average of daisies per metre squared. For me, this is 7 daisies per metre squared. To estimate the total population, I just need to multiply this number by the total number of metres squared in the field, 620. This gives me 4,340 daisies. You can use this method of random sampling to compare population sizes in different areas, or even repeat this for the same area at different times. However, there is another way of using a quadrat that allows you to observe how population density varies between 2 points within an area. For this, we are going to use a transect and a quadrat. The first thing I need to do is lay a tape measure along the area I'm going to be sampling. I've laid this one out to a distance of 10 metres. I'm then going to sample along this line at 1 metre intervals and count the number of organisms and record those results. So I place the quadrat at the start of my transect and I can see that there is 1 daisy on the edge of my quadrat here, so I will record that in my table. I then need to move the quadrat up the transect 1 metre at a time and count the daisies each time. Here are my results. I've plotted a line graph to show how the population density changes with distance from the gate. I could also use a kite graph. As you can see, the number of daisies per metre square increases the further from the gate we are. The most likely cause of this is people trampling them as they walk through the gate, the effect of which decreases with distance from it. This is what we call a biotic factor, as it is due to living organisms. Abiotic factors are those that are not due to living things. For example, light intensity or pH of the soil.
All right, so that was the quadrats, right? Anybody have any questions for the quadrats so far?
No.
All right, so we're gonna deal with line transects now. So let me get a line transect for you and let you see it. Have to play the video for you guys to understand this, right? Right. So these lines here are the transect lines. You have the boundary area of your study, you have your objects in your study, right? Now it's a horizontal line usually made of string that is placed across an ecosystem or parts of it. The species of plants and animals that are slow moving or stationary that touching the line at fixed intervals are recorded, right? Measuring the height of the line at each sample point will give a profile of the ground below the line. Now, these are actually used mainly where one habitat changes into another. So like if you have a grassland merging into a woodland, or for shrubs and trees layers in a woodland, this is good. This is actually pretty good for that particular type of sampling technique, right? So I'm gonna try to get a video with the line transect sampling method for you guys to see how it's done, the basics of it. And then we have to look at sweep nets and capture recapture.
So how do we count the residue? We've got our cord laid out, and if we see a black spot, we look, is there a piece of residue? Now, what is residue? It's any piece of plant material, living or dead, that covers the soil surface. So if we find something there, we're going to make a note of it. That's one. We look at our next black mark. There's no residue there, so we don't count. Well, here's residue, but there's no black mark, so we don't count. There's another black mark, and there's some dead plant material under it, so that's two. We go to the next black mark, and there's no material, so we don't count. So in this case, as we've looked at those first four marks, we found two pieces of residue. You do that for the whole 100 points on the cord, so 33 meters, about 100 foot. You do that, you take the count, and when you're done, what you have is the percent residue cover.
All right, any questions so far from anybody? No? All right, so we're moving on again. We're gonna look at sweep nets. Right, so it's basically a net that it does have, and they use this to sample flying insects, right? So the sampler will stand with the net and sweep the net a few times like this, right? In order to catch the insects, right? It's randomly done in a certain ecosystem. Each number of species collected in the net is counted, and then they're obviously released. No organisms are harmed during the sampling technique process. And these sweep nets could be actually used together with quadrats or line transects. So this is one that you could use hand in hand with another sampling method. So I'm getting a video for you guys with this one.
A sweep net is a cost-effective way to monitor for the presence of a variety of insect pests. Standard nets have a diameter of 15 inches. It's important to use a standard size net each time you sweep, so you are able to compare your results to others in your field. To start, hold the net with the hoop end nearest to the ground in front of you. Swing the net from side to side in a full 180-degree arc. Sweep one stroke per step as you casually walk through the field or down the road. After 10 to 15 strokes, shake all your contents to the end of the net. Close the mouth of your net. Have a Ziploc bag ready to go. Push inside out all the contents inside the Ziploc bag. Close the bag. And now you're ready to start pinning and labeling your collection.
All right, so that's basically it for that one, which is the sweep nets. The last sampling method we're going to talk about is the capture-recapture method. And this is used to estimate population sizes of animals that are moving, right? So it's a representative of sample animals of species that are trapped, counted, and marked. And after marking, they mark it with a spot of paint that's not going to harm the organism. And then the animals are released in the same area. And a day or more later, a second sample is captured and marked and counted as well, right? And they be able to calculate it. So the first sample, the number of organisms in the first sample is multiplied by the number of organisms in the second sample and divided by the total number recaptured. So I'm going to try and see if I can get a video with this one as well. Right? To show you guys how this one is done. This one is also known as the Lincoln Index. All right. So I'm going to give you guys some points here based on the sampling techniques. Just give me a minute. Let me pull it. Right. And then from there, we're going to talk about measuring abiotic factors a little bit, like climatic, edaphic, right? Edaphic, other ones like wind speed and stuff, and how you treat the results, right? Right, so when you're finished here, let me know. Guys, how far will you reach? I could scroll down. Mapping the ecosystem. So let's talk about collecting organisms, right? Now, to identify the organisms, they may be collected, but it's not really essential, right? They could be simply given code names, right? Organisms should not be collected or destroyed unnecessarily because of the fact that you could disrupt your ecosystem and you don't want to do that, right? So when you're collecting living organisms, please try and collect as little as possible and return them to the environment. Don't destroy them, don't harm them at any point, right? And try to not disturb their habitat as well. Again, you can throw off the ecosystem, right? So when we're looking at the abiotic factors now, to relate distribution and behavior of organisms to abiotic factors, you have to compare a couple of things. So like, for instance, climate, right? Certain climates will influence certain organisms, which we will have established. So like if you're measuring temperature, you could use a thermometer. Humidity, you could use a wet and dry bulb thermometer. If you want to measure light intensity, there is a photographic light meter to be able to determine how much light is present in an area. If you're looking at rainfall, there is the rain gauge you can use. If you're looking at factors that affect plant growth, like for instance, pH, universal indicator, there's being strips. You take your strip, you put it into your soil, and there are different color ranges for how acidic or how basic your soil content is. If you want to check the rock particles, right? Sedimentation, right? So you're shaking a fixed volume of the soil that you want to test for in some water, right? There's a measuring cylinder, and you just leave it to settle, and then you take your value readings from there. Water content, right? So you weigh a soil sample, then you dry it and you reweigh it again, right? You take the first weight minus the second weight, right? Divided by the first weight, multiply by 100. I'll give you the calculation just now. If you want to look at the humus content, right? You're heating your soil sample, right? To burn off any humus. You're weighing it, you're weighing it first, then you're heating it, drying off the humus. Same calculation again, multiply by 100 to get the percentage, right? Water holding capacity is what they always use. They always check this one. So you're placing a fixed volume of your dry soil into a funnel, plugged with a wet cotton wool, and you're pouring a known volume of water through the soil. And then you're collecting the water that drains through and you're measuring the volume. So I'm going to play a video for that to check for water holding capacity for you guys. But this one in your lab, when you're checking to compare the water holding capacity of soils, right? This particular calculation is always done. Yes.
Earlier this morning, we need to know how much water soil can retain. And we call it the water retention capacity of soil. It is important to the farmer because if the soil retains too much water, the soil will become waterlogged and plants won't grow well. Sandy soil, for instance, might let through too much water and the plants will dry out too fast. Loam, which is the type of soil that's mostly preferred in agriculture and in gardening, might have a in-between water holding capacity. In other words, just perfect for the plant's needs. And what I have here are 3 measuring cylinders, 100 milliliter each, 3 funnels, with filter paper and soil specimens. Let me just show you how one should fold the filter paper. Over in half, and then into quarters, but with a slight overlap. Open it, one part on the one side, put it in your cylinder. Okay. Now we have the same amount of soil in each of the funnels. I took 3 big scoops of soil. Of course, if you want to have exact measurements, you should weigh the soils to have the same amount of soil. And then I pour 100 milliliters of water into each funnel. This is the loam, the garden soil. And you can see the water is already started to drip through. The more clay soil and the sand. Right. Now this is going to take a while, so let's have a look at the samples that I did earlier this morning. Right, number 1, sand. Remember, it had 100 milliliters of water poured into the funnel. Number 2, loam. And number 3, clay. Perfect. Can you see that the sand let through most of the water, very low water holding capacity? The clay only left, if we read this carefully, about 42 of the 100 milliliters, and loam in the middle. And look at the color of the water that ran through the loam, dissolved salts, which also tells you and gives you an idea that this type of soil contains a lot of nutrients. So by comparing the amount of water that ran through, we can determine and compare the water retention capacity of different soil types.
Right, so that's basically how you carry out that particular lab. Like I said, this is one that you guys will have to do in school. I'm very, very certain of that, right? So it's real simple with that. Anyways, so what I want you guys to do here now for me is to take down these couple points. By that time you're finished with these couple points, we could wrap up. The treatment of results is really simple. It's here on the slide, so I'll send for you guys to take a look at. It's not really much, because it's basically telling you how you would actually do these labs. So it's not really much to go through with some examples. So I'll make sure and send this for you guys one time, all right? So once you're finished here, let me know. Take your time, because we have a couple of those factors. We have 3 factors that you have to take down, right? So let me know when you're finished.